Hello everyone, my name is Jakub Staroń and this is the second episode of scientific seminars called MedChem Essentials. The topic of today's presentation is permeability in a cellular context. On the last slide of previous presentation, we presented this chart that described how lipophilicity affects different aspects of drug design and development. And this chart will be a roadmap for us for the upcoming series. The first topic that we'll be discussing is permeability. To start talking about permeability, we need to introduce the concept of cellular membrane. Then we'll move on to description of different permeability mechanisms, efflux systems, and finally of membrane components. Membrane is a fluid-like bilayer arrangement of phospholipids. It exists in an intermediate state between a true liquid and true solid state, because the lipids and all the proteins that are embedded within the membrane, they have limited degree of rotational and lateral movement. The outer leaflet of this bilayer consists mainly of electrically neutral lipids like phosphatidylcholine and phosphatidylethanolamine, whereas the inner layer of membrane consists of negatively charged lipids like phosphatidylserine. This will have profound consequences on another aspects of drug design and development. Another components of membrane are proteins, and these are like peripheral and integral proteins represented by various receptors and transportation routes for different substances in and out of the cell. To be absorbed, a molecule needs to pass through the cellular membrane. And because of that, membrane permeability will be key determinant of absorption, distribution, and excretion. Membrane is primarily lipophilic in nature. And because of that, lipophilicity is the main characteristic that governs permeability. So more lipophilic substances will be better absorbed, better permeable through the membrane. Less lipophilic, more polar compounds will be less permeable. Now let's move on. Majority of marketed medicines are administered orally. Because of that, the main physiological barrier for these drugs will be the intestinal epithelial membrane. The permeation of drugs through this intestinal epithelial membrane can occur via different mechanisms, which are passive transcellular pathway, paracellular pathway, and active transport. We'll describe them in detail in a moment. Another concept related to permeability and epithelial membrane is tight junction. These cells, epithelial cells that form this barrier, they are tightly packed. They connected very tightly to one to another. And the space that is between the cells is called a tight junction. This is a very narrow 50 to 200 nanometer cleft. In our body, there exists a very specialized type of epithelial cells which have very exceptionally tight junctions, and they constitute the blood-brain barrier. Our body wants to protect the brain from exposure to any unwanted substances that may affect its function. So it is enveloped in a blood-brain barrier, a very tightly packed barrier, impermeable barrier. Now let's move to description of different types of transport. On this chart, we have a schematic representation of a cell and the tight junction. Primarily, compounds can be absorbed via a paracellular pathway A through these water-filled pores. That there is a restriction because of these tight junctions, because they are very tight, only hydrophilic and small compounds can be absorbed via these mechanisms. The limit is that the molecular weight of a compound cannot be larger than 200 Daltons, and this molecular radii of this molecule cannot be larger than 10 angstroms. Another mechanism is transcellular pathway. So in this mechanism, a compound needs to pass through one membrane and then through second membrane to be fully absorbed into the body. And the third mechanism through which compounds can be absorbed into the body is the active transport. Active transport exists so as to absorb nutritious substances. These nutritious substances are sugars, amino acids, proteins. They are very polar and often large molecules, so they cannot rely on being absorbed through paracellular pathway or transcellular pathway. So for this reason, there are specialized transporter proteins for sugars and amino acids and peptides that help to absorb all these compounds into the body. And we can hijack these mechanisms. There's a strategy to couple a drug with a sugar molecule or amino acid molecule, and thanks to that, our drug can be recognized by these proteins and be effectively absorbed into the body. But our body also has efflux systems, like protective mechanisms. It wants to protect itself from exposure to any toxins or harmful substances. Thus, in the outer layer of the membrane, there are efflux pumps, as very specialized proteins, that any substance that is being absorbed, that is potentially harmful, is being recognized by this protein and is being kicked out outside of the cell. These efflux systems are mainly composed of P-glycoprotein. This is the most abundant component of so-called multi-drug resistance in a variety of cell types. This P-glycoprotein, or called PGP, is a quite large protein that on an energy ATP-dependent way, it pumps out, fluxes out the compounds, wide variety of drugs. It recognizes really different substances. It's very promiscuous protein. Interestingly, the expression of MDR1 gene, this is a gene that encodes PGP, it is commonly seen in tumors that are treated with chemotherapy that relapse during the course of the therapy. And this is the main reason for drug resistance of tumors. Now let's move on to the description of different components of cellular membrane. As we described before, we have glycerol phospholipids like phosphatidylcholines, ethanolamine, phosphatidylserine, phosphatidylglycerol, phosphatidylinositol, some sphingolipids, and cholesterol. So that's it for today's presentation. Thank you very much and see you next time. Bye.